Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students. And as we have completed the syllabus, I intend to do the revision notes because this will help you a lot to understand what you have been taught, and this will be reinforcement of learning. And once we complete this revision notes, I'll be posting you different types of questions like MCQs of the chapter, CBSE question answers, and then third assertion and reasoning questions, HOTS, higher order thinking skills questions, all this, so that once you have the self analysis, you will be in a position to understand whether you are confident of that chapter. Once you are confident of that chapter, then you go for the next chapter, and this is how you need to understand. If there is any doubt in this chapter, I ask you to go for my video related to this chapter and clear your doubts. Further, still you have doubts, you can post doubts in the WhatsApp group. Please do that. Now the revision notes, we are going with the chapter 2. Long back we have done somewhere in the month of June, so it's a sort of memory. So the title of that is Sexual Reproduction in Flowering Plants, because first chapter is not there for your batch. We are going straight to the second chapter. So reproduction ensures continuity of species generation after generations as the older individuals undergo senescence and die. Senescence means old age. Flowering plants shows sexual mode of reproduction and bears complex reproductive units as male-female reproductive units along with accessory structures. Flower is a modified stem. What is a flower is known as? Modified stem, which functions as a reproductive organ. So flower is a reproductive organ and produces ova and pollen grain. A typical angiosperms flowering plants. Angiospermic flower consists of 4 whorls of floral appendages attached on the receptacle. So there will be 4 structures in the form of rounds, circles. One is calyx, another is corolla, third one is androecium, that is male reproductive organ consisting of stamens, and fourth is gynoecium composed of ovary, style and stigma. Let's see that. So this is clear. So you got now the sepals. Group of sepals is calyx, and petals, group of petals or corolla, so PC, and then you got stamen or anther, that's androecium, and this anthers consists of stamens and a stalk, and then gynoecium, which will be in the center, consists of 3, that is style, stigma and ovary. So this central portion which is there, that is style, stigma and ovary. So these are the 4 whorls. So what is the first one? First one is calyx, second one is corolla, third one is androecium, and the fourth whorl is gynoecium. So 4 whorls will be there. So pre-fertilization, before fertilization takes place, what are the structures and events? Several structural and hormonal changes lead to formation and development of the floral primordium. Inflorescence is formed that bears floral buds and then flower. In flowers, male androecium and female gynoecium differentiate and develops, in which male and female gametes are produced. So it is the gonads. So from gonads, gametes are produced through meiosis. So the first one, stamen, Microsporogenesis and pollen grain. Stamen consists of long slender stack called filament and generally bilobed anthers. Bilobed, 2 lobes will be there. Each lobe contains two theca, so we call it as dithecous. The anther is four-sided structure, consists of four microsporangia, two in each lobes. So this is how the structure will be. So this is the stalk, anther, and when we take this section of this TS, transfer section, you can see the pollen sacs, pollen grains, and of course line of dehiscence. So microsporangia develop further and become pollen sacs. So microsporangia are becoming pollen sacs. And pollen sac, as you are seeing, contains pollen grains. Microsporangium is generally surrounded by four layered walls. So the microsporangium will be having epidermis, epi means upon, dermis means layer, outer layer, endothecium, middle layer, and most important, tapatum. So the innermost layer, tapatum, nourishes the developing pollen grains. So normally this is 1 mark question in your CBSE. What is tapatum? What is the function of this? So tapatum is the innermost layer of the microsporangium. And what is the function of it? It nourishes the developing pollen grains. So nourishment, it provides nourishment. And sporogenous tissues, it is compactly arranged homogeneous cells which are present at center of each microsporangium when the anther is young. And microsporogenesis, this is also again 1 mark question, the process of formation and differentiation of microspores. So microspores, common name, pollen grains, from MMC, microspore mother cell, by meiosis. It's called microsporogenesis. So formation of microspores. Microsporogenesis is formation of microspores. From where? From microspore mother cells. How? Through meiosis. So the cells of sporogenous tissues undergo meiotic division to form microspore tetrad. Tetrad means 4. As the anther mature and dehydrate, the microspore dissociate and develops into pollen grains. So this is the section. So you got epidermis, outermost layer, endothecium, the second layer, inner layer, and then you got middle layers, and then most important one is the tapatum, the nourishment, and below the tapatum you can see microspore mother cells, MMCs, and these MMCs will form microspores, pollen grains. So when you are doing this revision notes, you must keep pencil and paper ready, draw the diagrams, label it, understand the significance of each label, then only you can move to the next page. So this is the tetrad. You can see the pollen grains, dehiscence. Pollen grain represents male gametophyte. Pollen grains are made up of 2-layered wall. And pollen grain, each pollen grain will have 2 layers, exine and intine. So it is sporopollenin. Exine is made up of sporopollenin, the most resistant organic matter known. It can withstand high temperatures and strong acids and alkali. No enzyme can degrade sporopollenin. That is the greatness of this. So sporopollenin, exine. And intine, it is thin and continuous layer made up of cellulose and pectin. And in between you got germ pores. These are pores, apertures on exine where sporopollenin is absent, and that forms pollen tube. The germ pores will allow that. And a plasma membrane surrounds cytoplasm of the pollen grain. Now, mature pollen. A mature pollen consists of 2 cells with a nucleus. So one is vegetative cell, another is generative cell. So vegetative cell is big, abundant food resource will be there, large irregular nucleus will be there, responsible for the development of pollen grain. And generative cell, compared to vegetative, it is small. It involves in syngamy, fusion with egg, and then dense cytoplasm and nucleus. As you are seeing, you can see vegetative cell is big, so this much portion, and the generative cell is small, and it has got a dense cytoplasm with the nucleus. So pollen grains of many species like Parthenium cause severe allergies and bronchial diseases in some people and leads to chronic respiratory disorders like asthma, bronchitis. That's why pollen grains are allergic to some people. Pollen grains are rich in nutrients and are used as pollen tablets as food supplements. This is the positive side of the pollen grains. So viability of pollen grain varies with species to species and should land on stigma before this period to germinate. Pollen grains of large number of species are stored in liquid nitrogen. Look at that, liquid nitrogen at temperature minus 196 degrees called pollen banks. So we have got pollen banks. And then continuing with the female, the pistil, Megasporangium, there it is Microsporogenesis, here it is Megasporangium and embryo sac. So gynoecium may consist of single pistil. If it is single pistil, we say monocarpellary. More than one pistil, polycarpellary, which may be fused. If it is fused, syncorpus. If it is free, apocarpus. These are the technical words that you must write during your practicals. So multicarpellary, syncorpus pistil like papaver. Multicarpellary, apocarpus means free, then mycelia. Each pistil has three parts, as we said just now: stigma, style and ovary. Inside the ovary is ovarian cavity, we call it locule. The placenta is located inside the ovarian cavity. Megasporangia or Megasporangium, ovum or ovules, arise from placenta. So Megasporangium, how it is formed. So ovule is a small structure attached to placenta, and funicle, stalk by which ovule is attached to placenta, that is funicle. Hilum, junction between ovule and funicle. Integuments are protective envelopes, and micropyle, small opening at the tip of the ovule into where pollen tube enters. So these are all the technical words that you need to remember. And here you can see chalaja, basal part of the ovule. Nucellus, 2n, mass of cells enclosed in integuments, has abundant food reserve. So this is the structure, and you can see the down from chalajal pole, embryo sac, nucellus, inner integument, outer integument, micropolar pole, micropyle and funicle, hilum. So draw the diagram, write all the labeling and understand the importance of each one. And we go for the Megasporogenesis.